The gut-brain connection, how your microbiome affects neurological health. Welcome. This is the official GlobalRPh YouTube channel. If you haven't already, hit that subscribe button to stay updated with our latest videos. Whether you're looking for medical insights, the latest healthcare trends, or fun educational content, there's something here for you. Abstract. Emerging research has increasingly highlighted the gut microbiome as a major player in neurological function and overall brain health. This review delves into current scientific evidence exploring the intricate relationship between the gut and the brain, collectively known as the gut-brain axis, and examines how microbial communities in the gastrointestinal tract influence neurological processes. Recent studies have shed light on how gut bacteria interact with the immune system, neuroendocrine pathways, and the central nervous system, CNS. These interactions are far from passive. They actively shape brain development, modulate behavior, and may contribute to the onset or progression of various neurological and psychiatric disorders. Key findings from the literature highlight several mechanisms through which gut microbiota exert influence on the brain. Neurotransmitter synthesis and signaling. Certain microbial species are involved in the production of key neurotransmitters such as serotonin, dopamine, and gamma-aminobutyric acid, GABA, which are essential for mood regulation, cognition, and sleep. Regulation of neuroinflammation. Dysbiosis, or microbial imbalance, has been linked to increased systemic and neural inflammation, a known contributor to conditions such as depression, multiple sclerosis, and Alzheimer's disease. Modulation of the hypothalamic-pituitary-adrenal HPA axis. The microbiome appears to influence stress responses through the HPA axis, potentially affecting the onset and severity of stress-related disorders such as anxiety and post-traumatic stress disorder. Furthermore, accumulating evidence supports a role for the gut microbiome in the pathophysiology of neurodevelopmental and neurodegenerative diseases, including autism spectrum disorders, Parkinson's disease, and Alzheimer's disease. These associations are being investigated not only for their diagnostic value, but also for their potential to inform novel therapeutic strategies, such as probiotics, prebiotics, dietary interventions, and even microbiota transplantation. By synthesizing the latest findings, this review aims to equip healthcare professionals with a comprehensive understanding of the gut-brain axis, its biological underpinnings, clinical relevance, and future research implications. As our knowledge of the microbiome expands, it opens new frontiers in neurological care, emphasizing a more integrated approach to mental and cognitive health that includes the gut as a central therapeutic target. Introduction. The gut and brain are intimately connected in ways we are only beginning to understand. Long regarded as separate systems, emerging research shows that the gastrointestinal tract and central nervous system engage in continuous, bidirectional communication, forming what's now recognized as the gut-brain axis. At the center of this communication is the gut microbiome, trillions of bacteria and other microorganisms residing primarily in the colon. Their influence spans far beyond digestion, extending into brain development, neurotransmitter synthesis, immune modulation, and even behavior. This video explores the most compelling mechanisms linking the gut microbiome to neurological health and disease, summarizing current findings relevant to physicians and scientifically engaged readers. The gut-brain axis, anatomy of a conversation. The gut-brain axis operates through a complex network of neural, endocrine, immune, and metabolic pathways. The vagus nerve, a rapid neural link. The vagus nerve is the primary neural pathway facilitating communication between the gut and brain. It senses microbial signals and relays them to the brainstem and higher cortical areas, influencing mood, pain perception, and stress responses. Immune pathways, inflammation as a mediator. Approximately 70% of immune cells reside in the gut. Microbial balance helps maintain immune tolerance, while dysbiosis, an imbalance in microbial populations, can trigger systemic inflammation and increased intestinal permeability, leaky gut, both of which have been linked to neuroinflammation seen in conditions like depression, Alzheimer's disease, and multiple sclerosis. Endocrine signaling. The HPA axis. The hypothalamic pituitary adrenal HPA axis, central to stress regulation, is also influenced by the gut microbiome. Gut microbes can impact cortisol levels and stress reactivity, with implications for anxiety, post-traumatic stress disorder, PTSD, and depression. Neurotransmitter and metabolite production. The gut microbiota participate in the synthesis and regulation of key neurotransmitters and bioactive metabolites. Serotonin, nearly 90%, is produced in the gut. Certain microbes stimulate its production by acting on enterochromaffin cells. GABA, lactobacillus and bifidobacterium species can produce this inhibitory neurotransmitter involved in calming neuronal activity. Dopamine and acetylcholine, microbial precursors and enzymatic modulators, influence these neurotransmitters critical for motivation, reward, and cognition. Microbial metabolites such as short-chain fatty acids, SCFAs, including butyrate, propionate, and acetate, support blood-brain barrier integrity and regulate neuroinflammation. Other byproducts like tryptophan metabolites and bile acids can also shape brain signaling and behavior. Gut microbes in neurological and psychiatric disorders. An expanding body of research has linked gut dysbiosis to a wide range of brain-related conditions. While causality is still being untangled, the associations are compelling and warrant clinical attention. Depression and anxiety. Alterations in the gut microbiota composition have been observed in individuals with depression, often marked by reductions in lactobacillus and bifidobacterium. Preclinical studies show that transferring gut bacteria from depressed humans to germ-free mice induces depressive-like behaviors in the animals, suggesting a causal microbial component to mood disorders. Autism spectrum disorder, ASD. Children with ASD frequently suffer from gastrointestinal disturbances, prompting investigation into the gut-brain relationship. Several studies report altered microbial diversity and abundance in ASD populations. Though early, trials of microbiota-directed interventions, like probiotics or fecal microbiota transplantation, FMT, are showing potential to alleviate some behavioral symptoms. Neurodegenerative diseases. Microbiome alterations may precede or exacerbate conditions like Parkinson's and Alzheimer's disease. Parkinson's disease. Microbial shifts have been documented before motor symptoms arise. Alpha-synuclein aggregates in the enteric nervous system hints at a gut origin hypothesis for disease onset. Alzheimer's disease. Certain gut bacteria may influence amyloid plaque formation through inflammation and metabolite production. SCFAs and endotoxins have been implicated in modulating brain aging and pathology. Targeting the microbiome. Therapeutic potential. As understanding grows, microbiome-modulating therapies are gaining traction in clinical research. While most approaches remain investigational, the potential for adjunctive or even primary treatments is becoming clearer. Probiotics. Administering beneficial live microbes via supplements or fermented foods may restore microbial balance, reduce gut inflammation, and influence brain function. Early studies show promise in improving mood, anxiety, and even cognition, though effects are strain-specific and often modest. Prebiotics. These are non-digestible fibers that promote the growth of beneficial bacteria. By shaping microbial ecology, prebiotics may help modulate systemic immune responses and neurotransmitter production. Early trials are exploring their impact on depression, anxiety, and mild cognitive impairment. Diet. Nutritional interventions are one of the most accessible ways to alter the gut microbiome. Diets high in fiber, polyphenols, and fermented foods promote microbial diversity, whereas processed foods and high-fat diets are associated with dysbiosis and inflammation. The Mediterranean diet, in particular, has been linked to cognitive resilience and reduced risk of depression. Fecal microbiota transplantation, FMT. FMT involves transferring stool from a healthy donor into a recipient's gut to restore microbial balance. Originally developed for recurrent C. difficile infections, FMT is now under investigation for neurological indications, including Parkinson's disease, ASD, and multiple sclerosis. While initial results are intriguing, FMT remains experimental outside of infectious disease settings. Challenges and limitations. Despite exciting findings, several limitations hinder the translation of microbiome science into routine neurological care. Complexity and individual variability. Each person's microbiome is unique and influenced by countless variables: diet, age, genetics, medications, making broad generalizations difficult. Short study durations. Many studies are small, short-term, or conducted in animal models. Longitudinal human trials are needed. Methodological differences. Lack of standardization in microbiome sampling, sequencing, and analysis impairs reproducibility and cross-study comparisons. Placebo effects and hype. As interest grows, so does the risk of overpromising benefits without sufficient evidence. Rigorous clinical validation is essential. The road ahead. The gut-brain axis offers an exciting and still largely untapped frontier in medicine. With continued research, microbiome-based strategies may one day offer new hope for patients with depression, neurodegenerative disease, and other CNS disorders. In the meantime, clinicians can begin to consider gut health as a relevant factor in holistic neurological care. Future directions include long-term cohort studies tracking microbiome composition and neurological outcomes over time. Identification of microbial biomarkers for early diagnosis or risk stratification. Development of precision probiotics tailored to individual microbial profiles. Investigations into how lifestyle factors, stress, sleep, physical activity, interact with gut microbes and brain function. Conclusion. The gut-brain connection represents an exciting frontier in neuroscience and medicine. The evidence presented in this video highlights the significant influence that gut microbes can have on neurological health, from neurotransmitter production to inflammation regulation and potential impacts on various neurological disorders. For healthcare professionals, this emerging field offers new perspectives on the treatment and prevention of neurological conditions. While more research is needed to fully understand the complexities of the gut-brain axis and develop effective therapies, the current evidence suggests that considering gut health may be an important aspect of neurological care. As our understanding of the gut-brain connection continues to grow, it has the potential to revolutionize our approach to neurological health, offering new hope for patients and new tools for healthcare providers. Thanks for watching our GlobalRPh production. If you enjoyed the video and found it helpful, don't forget to smash that like button and subscribe to our brand new YouTube channel. We're bringing you a dynamic mix of content across a variety of topics, so there's always something fresh, engaging, and insightful just for you. Your support means the world to us. It helps us grow and keep delivering exciting, high-value content. Stay tuned, because the best is yet to come.